Hi everyone and welcome again. In the last video, I introduced you to nucleic acids. Let me just remind you a little bit about them. First, nucleic acids store, transmit, and express the genetic information in the living cells in living organisms. And there are two main types of nucleic acids, DNA and RNA. And we mentioned that we may think of DNA as the book of life, where all the chemical instructions for making a living being is stored. And think of RNAs as the helper molecules, which take some of the information and instructions stored in DNA and transfer them to special factories to make and build the proteins needed for all life functions. Now, let us go to some more details about nucleic acids. Nucleic acids are very long and large molecules, macromolecules, composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus atoms, and made up of monomers called nucleotides. Nucleotides are joined to each other by covalent bonds, giving the molecule the form of a long polymer, long chain. Nucleotides are made up of three subunits: one or more phosphate groups, a pentose sugar, 5-carbon sugar, either deoxyribose or ribose, and a nitrogen-containing organic base. Nucleotides are joined next to each other by chemical bonds to form a polynucleotide chain, the basic form of nucleic acid. Now, let us become more acquainted about the amazing DNA molecule and talk about its nature, structure, and main roles and functions needed for life. Why we may think of DNA as the book of life? We as living organisms are living buildings that were made according to a mysterious plan, having inherent laws and rules for controlling and regulating the life processes in order to maintain life and ensure the survival of the living organism. So, cells in our bodies contain DNA, which defines the general plan and necessary information and guidelines for making and developing a human being. So DNA, as the blueprint of life, has the plan or recipe and instructions needed to make the living organism and maintain its life. DNA instructions are written in an amazing specific language, the genetic language. English has about 26 letters. They are used to write almost infinite numbers of words, phrases, and sentences. Likewise, the genetic language has 4 letters, and they are used to write volumes of tremendous numbers of words, sentences having different genetic meanings, which are translated at the end to making and keeping the living organism, as we shall see during the course. So DNA carries all the genetic information responsible for all the traits specific to each organism. In humans, these traits include all the seen or unseen traits, such as the color of the hair, the skin, the body dimensions, immunity to diseases, predisposition to genetic diseases, and so on. I remember that we were taught in school years that chromosomes are the carriers of genetic information. So what is the difference between DNA and chromosomes? Very simple. The chromosome is composed of one DNA molecule wrapped in a very unimaginable, complicated, and compacted way around proteins called histones. This complicated wrapping is important for condensing the very, very long DNA molecule inside the very, very small nucleus in order to protect it and keep it safe and be able to regulate its functions. In order to understand the wondrous workings of DNA, we first need to look at its structure, because structure determines to a large extent the functions of this amazing molecule and the way it performs in our lives. DNA is a very long polymer containing the monomers called deoxynucleotides. The deoxynucleotide is composed of a nitrogenous base, a deoxyribose sugar, and a phosphate group. The deoxyribose sugar is ribose sugar lacking the oxygen at the carbon number 2'. There are 4 nitrogenous bases present in DNA. They are called adenine, guanine, cytosine, and thymine. Adenine and guanine are the double-ring purine bases, and cytosine and thymine are the single-ring pyrimidine bases. After getting acquainted with the nucleotide units of DNA, let us see how these units are combined to generate the general shape of the DNA molecule. DNA has the shape of a double helix, composed of 2 polynucleotide strands intertwined around each other to form the helix. Each DNA strand is a polymer of deoxynucleotides linked to each other by covalent bonds between the deoxyribose sugar and the phosphate group. This bond is very specific and is called the phosphodiester bond. So, the deoxynucleotide units in the same strands are connected by strong covalent bonds, the phosphodiester bonds. But how the 2 strands are connected to each other to make the helix? Let us see. The nitrogenous bases in one strand are linked to those in the other strand by hydrogen bonds. Hydrogen bonds are weak bonds, non-covalent bonds, and play a very important role in shaping the molecules of life. So, the 2 strands are held together by hydrogen bonds formed between the bases. Adenine and thymine are paired together through 2 hydrogen bonds. Guanine and cytosine are paired together through 3 hydrogen bonds. This is the rule of base pairing, by which an adenine in one strand binds or pairs with thymine in the other strand, and a guanine in one strand pairs with cytosine in the other strand. The base pairing rules makes the 2 strands of DNA complementary to each other, meaning a G, guanine in one strand, is linked to a C, cytosine in the other strand, and an A in one strand is linked to thymine T in the other strands by hydrogen bonds. A question might pop in our minds. Why weak hydrogen bonds are used to make the helix? Why not use strong bonds? Even though hydrogen bonds are non-covalent weak bonds, they are present in thousands in a DNA molecule and contribute so much to the stability of the double helix. As they say, power is in unity. Also, being weak bonds, this makes DNA helix more flexible and ready to change its shape under certain conditions, as we shall see in DNA replication. So, let us talk about DNA replication. What is the significance of DNA replication? To maintain life and preserve the species, all cells in the organism must contain identical copies of the genetic material DNA. Whenever a cell divides, there is a need to synthesize to make 2 identical copies of each DNA molecule present in the original cell. This is to ensure that each new cell receives an exact copy of the DNA which was present in the original cell. For example, in humans, before a cell divides, all 23 pairs of chromosomes are replicated to form 46 pairs, so that following cell division, each daughter cell takes a full set, 23 pairs of chromosome, identical pairs of chromosome. Now, let us have a general look at the DNA replication process, which is vital for our existence and for life. During DNA replication, each parental strand acts as a template for the synthesis of the new daughter strand, which is complementary to the parental strand. By the action of specialized protein molecules, the two strands in the double helix separate away from each other to allow the reading of the nucleotide sequence in one strand and use it as a template to make a new complementary strand. Let us take an example for this situation. For example, if the parent strand has a T, thymine, the new strand will have an A, adenine. If the parent strand has a C, cytosine, the new strand will have a G, guanine. This results in making two new molecules of DNA having the exact nucleotide sequence present in the original DNA molecule. DNA replication is mediated by a group of protein molecules. The most notable is the enzyme DNA polymerase, a hard-working enzyme, which is responsible for linking nucleotides next to each other to make the DNA strand. DNA polymerase doesn't work alone, but gets the help from other enzymes and proteins, which cooperate to execute the DNA replication process with an astonishing level of accuracy. Why accuracy? Accurate DNA replication maintains the genetic integrity and the faithful transfer of genetic information between cells and from one generation to the next generation. This faithful transfer of genetic information is essential requirements for life to start and continue. And this is a very simplified image and example expressing the beauty of the hidden microscopic world in our bodies, the amazing microscopic world. Thank you for watching, and see you in next videos to delve together into the amazing world of the molecules of life. Your questions and comments are always welcome. Goodbye and see you later.